Okay, well, welcome everybody to the sixth lecture out of 14 of E443. Today, Josh has agreed to give the presentation, and we're going to start out with a review of people's projects as usual, and also of homework. And then we're going to jump in and do the implementation and meaning of chemical diffusing fields and do some examples of that. And so, without more ado, I'll turn it over to Josh and let him steer.
Um, hello, everyone. As James mentioned, I will be talking about how to represent concentration fields and how to make these fields diffuse in CompuCell. Just feel free to jump in if you guys have any questions, or if you need me to repeat something, or if I'm going too fast, just feel free to interrupt me. So as a quick overview of what the lecture is gonna be, as James mentioned, we're gonna start with a summary of or an update on your projects. That this part is really important. We wanna know that everyone is making progress and help people if they are experiencing roadblocks. So we just wanna check in with you. Then we were gonna go over a little bit of what we talked about last time about how to conceptually think about models, how to break them apart into their basic components, and how to think about the interactions and the dynamics of these components. We'll talk about how what a good workflow for model development would look like once you're able to break your model apart into its conceptually into its different components, how you can go from that conceptual model into an actual implementation in a computational implementation and a simulation of the model. We're gonna continue talking about cell growth and cell division. And we're gonna take it from where we left it at last time, how to control cell growth using pressure. We'll talk a little bit about its meaning in its proper biological context. And this is gonna help us to review some of the issues that some people might have experienced in the homework, specifically how to measure pressure in the simulation domain, how to average pressure over different cells, and so on. After we review cell growth, we're gonna jump into concentration fields, talked about different transport processes, and then we're gonna do a couple of exercises on diffusion, and we'll come back to that later. So James, if you wanna direct here the updates on the student project.
Okay, well, I know we had an assignment due this week, which was to try to do a plan for your projects. And I'm going to try this idea of trying to ask people to have equivalent of 2 homework problems worth of work on a weekly basis on the projects to help people stay on a rhythm of being productive. It's a bit of an experiment. Something that I wanted to say also about the homework problems is that if you find ways of improving the homework problems, please feel free to make those suggestions. I sent an email out to that effect. If you catch an error in a homework problem, or you have some rewording and those are significant, we'll definitely include those for next year. And I'm happy to give an extra couple of points of extra credit for a problem if you make a significant contribution to improving it. So I just wanted to say that. So maybe we'll quickly go around the room. Tyler, do you want to give us a quick update?
Sure. So Once again, I'm doing a model of CD8 T cell production in an early immune response. And my goal is to replicate the results of the paper. And the model in the paper looks to describe the differentiation and predict how cells are going to behave along their signaling pathway. And for next week, some of the milestones I have laid out for myself is I want to implement all the different cell types that I have in the model and like their attributes and behaviors. And I want to, using the volume constraints and the contact energy plugin. And additionally, if I have time, I'd like to implement the intracellular ODE model for my project, depending on where I get with the first milestone. And that's kind of where I'm at right now.
Okay, that's great. I think you're working with TJ, right?
Correct.
So we haven't covered integrating SPML and antibody models into CompuCell yet. That'll probably be another week or two before we get to that in the lecture. But it's not that complicated, and so TJ or Josh and I can help with that if you need help. So that's great. Okay, Devin.
Yeah, so for my cancer stratification model, I've been continuing to implement the existing CompuCell code, and I've done a few different sort of sub-projects, just implementing the base code. I haven't got to the glucose limiting factor yet, because I assume we'll cover that today in fields. And so, yeah, I've just been testing some of my results with like the cell field and some different cell counts, and everything's looking good so far.
Great, thank you. People, if you want a screen share, just say so. You don't have to today, but going forward with the little projects, it may be good to have one slide or so to show just as a part of your updates. I'm not requiring it, but I think it's helped. The discipline of making one slide to show a picture is not a bad thing. I don't want it to be burdensome to make. I don't want people to spend a lot of time making a whole slide deck for every week, but having one slide or two to show off what you've got, that might be not bad use of your time. Jeffrey.
Yeah, I'll share my screen.
Everybody sounds a little sniffly today. It must be spring.
Yeah. So this is just kind of overview of the end result. You see we have these convection cells here, and it's due to chemical reactions here in this square here. So I put the outline in here just to see it. So I'll work on making better convection flows this coming week, and also, well, I have slides for these things for this next week. So, and then I kind of, towards the end was a little bit vague because I like to keep things a little bit flexible. I don't know what'll be going on in a few weeks, but this is the main idea. We'll work on forces. And I'll talk to Andy a little more about this. So we're doing, right now we have the no slip boundary condition, and there might be an issue of the particle density at the boundary. They kind of accumulate there. So we're going to implement a potential so that some of them will approach it and not just to kind of eliminate the accumulation. But if they have a lot of energy, they'll go through the potential well. And then here is the cavity flow. You can see it kind of is moving more quickly up here, and then as it goes down here, it's more slowly, but still kind of following this path. And I need to work on the plots for this. This is just from CompuCell. But the simulation is working pretty well. I just need to get the plots. So yeah, that is what I have.
It's very impressive. I think once you could do those averaged velocity plots, you'll have a really good way of displaying it to show the Poisson velocities and the cavity flow. Those are great demos. And one could go through any of these standard fluid dynamics textbooks, and there are all these simple boundary flow near an obstacle of various shapes you could do. Flow around a sphere, a circle, flow around an airfoil, all of these, and they get the velocity profiles because those are mostly analytically calculated.
Yeah, there's one interesting flow that had like called the Stokes Pro with the oscillating plate. I wanted to look at. That might be kind of interesting. I forgot to mention it to Indy yesterday, but maybe we can talk about it later this week or something.
Yeah, that does sound interesting, and I'll have a new version out that has that gridding feature, so you can give it a grid size. Now then you can grab the velocity for grid locations tonight. So
okay.
That's great. And again, the point of these schedules is, I understand that, I think as I wrote in the homework, that people's schedules may change week to week a little bit, but having an overall rhythm, I think, is helpful. And that's why I wanted people to sort of have a plan this week, and then week by week you can update the plan as you see how you go, whether it goes faster, slower, new things happen. That's fine. Thank you very much. That's great. Fanbo, I think you're next on the list.
Yeah, so let me just share it. Okay, so basically in this week, I will implemented like to use the widget to rotate the camera of the simulation, and I can very quickly show you how this would work. So basically, if you change the angle, it can automatically switch the camera angles for that, and if you want to change back, so it will change back, and it works even if it's like you are running the simulation and you want to change the angle, it can be changed angle, and also it was still like in running the simulations. For next week, I will try to use a library called the PR event. I'm trying to basically create a very simple widget to use the mouse and use the keyboard to interact with the widget. Yep, I think that's for the next week's work.
Okay, well, that's fine. I'm impressed. Using the sliders or typing the angles isn't too bad. I know Jeffrey last semester did some 3D plotting using Matplotlib, and then had a slider that allowed you to rotate the 3D image. And that was actually not a bad interface. So if hover over with mouse winds up being too difficult, I think having slider controls would probably be enough. That's very impressive. Thank you. Okay, let's see who's next. Kali.
Can I share my screen because I have some code? All right, so my goal for this week was to basically create a skeleton model for Dictyostelium discoideum. It's not completely finished yet because I need to first make a custom initializer for this particular problem. So right now I just kind of have a blob as a placeholder. And I still need to implement the chemotaxis, which hopefully when we're discussing diffusion fields today, that can be of some help. So I'm looking forward to that. But basically I have initialized all the cell types and their contact energies. Something tells me I won't need a medium in this simulation. And then I've also implemented their target volumes and set up and arranged it so that cyclic AMP and ammonia are both going to be in the chemical environment, but obviously I still have to set up the exact chemotaxis for it because it looks like the way the paper models it, cells move towards greater concentrations of cyclic AMP, but the way I saw it in CompuCell, it wanted the chemotaxis to go towards another type of cell, so I just kind of wanted to see how I could implement that. So this is a skeleton. The purpose of making this was basically just so I could have some code down that I can then transfer over when I make my real model with the proper initialization and the chemotaxis and everything. To involve the context plugin, the volume plugin, and the chemotaxis plugin. And I think I added mitosis in there as well because I assumed that there was going to be some cell divisions. And I did have a few questions for you about this model specifically, but that can be arranged in another meeting because I don't know if we really have time to get into all that right now.
Okay, we should definitely, if you have time, I know you're very busy, but if you have time to set up a meeting for an hour later in the week, maybe Josh and I could sit down. I mean, one of the challenges is that we're presenting the material in some order. We have to present it in some order. And different people need different modules in CompuCell in different orders. And so we'll talk about diffusion today, but we probably won't get to chemotaxis till next week.
Okay.
But we can certainly sit down with you and explain chemotaxis this week. It's not that complicated. And so that sounds great. So I'd be happy to sit down with you. Let me know what your availability is Wednesday, Thursday, Friday, and we'll try to find it.
Thank you.
Okay then. Drew, I think you're the last.
Okay. Yeah. So my project is on that ovarian cancer drug delivery route paper, and since basically they're looking at two different models and comparing the two, I for next week at least, I think I'm going to try to set up. I'm trying to mimic the results in CompuCell, so I think for next week I want to get some initial setup for one of the models, and then probably the other the week after that. But not everything I know how to do yet, but I know for next week I'll at least be able to implement cell diffusion and stuff. And I think I'm probably also due for a meeting to figure out some of the other stuff. But also, you did link me the email to one of the people who worked on the paper, and I'd like to get in contact with them too.
But again, I mean, I send out these invites saying I'm available to meet with you. I can't force you to meet with me, but Josh and I are eager to help you out. So please, I know you're all very busy, but please do take advantage of that. We're happy to spend an hour going over these and covering things that maybe we don't get to in class until a little bit later. So if you want to meet with me and Josh later in the week, send us some times and we'll do our best to find an hour to sit down with you. Okay.
Thanks.
Okay, great. Okay, back to you, Josh, then. Thank you, everybody. That sounds great.
Yeah, and I do want to accentuate on what James said at the end. If you guys have questions, feel free to reach out and we're more than willing to help. Last time we talked a little bit about how to think about models in a conceptual way, how to break them apart into their components, and how to think about how those components relate to each other, and how those relations evolve in time and determine the state of the model, right, or of the object of the components of the model. And we stated that the model can be understood as being composed of four basic elements: objects with their properties and states, behaviors and interactions, dynamics and events, and initial and boundary conditions. By objects, we mean those components of the model that represent actual physical objects. Let's say in our case, cells, but they can also represent objects that might not be discrete, that might be continuous. For example, a field. Let's say we will talk a little bit about this later today, but let's say we want to represent a molecule as a chemical field that has different continuous values inside of or across or relates to other components of the model, depending on its continuous values across, let's say, a specific domain. And then the state of these objects have state that determine the properties of the objects dictate what their states are, right? Let's say the cells have a particular location, they have a particular volume, they have a particular surface. A field has, let's say, values that are associated with particular locations. And those are the states of the objects of the model. These objects, right, interact between each other, and those interactions are also part of the model structure. So for example, cells relate to each other. One cell can be the mother or the daughter of another cell, right? You have some sort of relation between them. Cells can also push against each other, right? And those are relational interactions, right? Those refer to how one object of the model affect a different object of the model. But also they might have intrinsic behaviors, right? Things that they do by themselves without or in disregard of their relation with respect to other objects. So for example, cells can grow, especially when we have, in those simulations where we have had one cell in the simulation domain and we've had it grow. So in that case, the cell volume, which is a property of the cell, it's increasing in time, right? So as a behavior, we could specify that the cell grows, right? Or the mass of the cell increases. So you can think of these objects as having these properties that dictate what type of behaviors they can engage in, and objects, different instances of these objects, or even different types of objects can relate to each other, and those relations are dictated by the different interactions that correspond, that are dictated by the model structure. Then if we look at a snapshot of what these properties are at a particular time, and what the interactions are and the behaviors are, we're, let's say, looking at a single state of the model, but the state of the model changes in time, right? Because these interactions are going to change. The interactions between the model components are going to change the properties of the model. Of the interactions are going to change the properties of the objects, and so on. And how the object properties and the interactions change in time is dictated by the dynamics of the model and by specific events. And we usually establish this distinction between, because by dynamics, we usually mean changes that are due to the model properties itself. You set up what the model is, how their how the model components interact between each other, and then the model behaves in a certain way. And events we usually refer to as discontinuous changes, as changes that occur, let's say, at a particular time, and they can sort of some often being imposed externally from the model. And finally, we to specify to fully specify a model, we need to specify what happens at the beginning, right? Of what is the initial state of the model, and then what happens when the model dynamics reach a certain condition. Let's say when in the simulation domain, for example, what happens to a cell when it reaches the boundary, or as we simulate the model, what happens when it reaches the end of the simulation. So and those are the specific implementation examples, but in general they refer to the initial and boundary conditions of the model. A very important aspect of this type of models, especially mechanistic models such as the one that we build here, is that in our models, nothing exists unless we assert it. And we've seen that in our examples when we assign a cell a particular volume, but we do not assign any surface, right? The shape of the cell we do not get for free the shape of the cell. To specify the shape of the cell, we need to specify both the volume, but also a specific surface tension. And we've talked about that and we've seen it multiple times in our homeworks. The reason we wanted to introduce that way of thinking about models is because by thinking about models that way, when you're working on your projects, you can start to think about how to decompose the model into its basic components, and then how those components can be represented in CompuCell specifically, but computationally more generally. Additionally, we propose a workflow that takes you from certain particular biological observations and problems all the way through choosing a implementation and simulation framework. The workflow starts with certain biological observations that motivate certain questions and hypotheses. So when you're reading the paper, if you choose to replicate a paper, try to think on how you answer, or how would you break down the development of the model in the paper, so it would fit into this workflow. And we have a unfinished paper that we worked for a long time, which was intended to take a paper that was already published, and then following this workflow, show that we can replicate that this workflow can ensure that you replicate the results of the original paper. But the idea is, at least at this stage of your projects, the idea is start by looking at what are the biological observations that motivate the project, right? Motivate the paper, motivate the model. What are the initial questions that the authors are asking themselves, and what are the possible hypotheses that they're going to use the models, that they want to use the model to test. Once these have been defined, then you can propose what a qualitative verbal model would be, using the diagram that we just explained, by breaking apart what the objects are, what the interactions would be, and how the properties and interactions change in time. And then from then on, you can quantify this by choosing a specific mathematical form to represent these properties and interactions. And then by identifying which are parameters of the model that are, and then by quantifying the parameters of the model, either be them independent or be model methodology, model and framework specific. So for example, we know that cells have a volume, right? Like real cells have a volume. And in terms of the quantitative model, right, the volume of the cell is a property, and how much of that volume we have is the state of that property, right? When we determine the method independent parameters, we talked about how to quantify that volume in a way that it can be mathematically represented, right? Let's say you have a equation that describes the growth rate of the volume of the cell, right? You start with certain volume, and then you have a growth rate, and then that determines what the volume of the you basically have a model of the growth rate of the cell. Now, let's say you choose to represent this model using CompuCell. So we know so far that CompuCell represents the model using the volume constraint, right? That has a lambda volume and a target volume, right? So those ways of thinking about the volume are method specific, right? Because they depend on the specific way that you decided to represent this property. And that is the main, and I've always sort of like struggled between that difference between what is the independent model parameter and the method specific parameter. But in general, for if you want to use this workflow to orient your work, think about how you will represent it generally in a mathematical form, and then how when you choose a specific framework, how do you what are the parameters that like emerge from the framework. And then once you've chosen a framework or a modeling methodology, then you can implement that methodology as a computational model and simulate it. And again, if people want to, for example, if people want to practice with their papers, the papers they have chosen to replicate, how to think in this in terms of this workflow, please do reach out, and we can just read the papers and okay, let's say and break it apart into this in this workflow. Okay, so last time in terms of coming back to cells and CompuCell, we talked about cell growth and division, and we said that we are not going to model the cell cycle explicitly yet. And someone mentioned that they wanted to represent started to work on the intracellular representations. Since you guys have done already cell growth and division, I don't remember who exactly it was, but I have a simple model that can represent the cell cycle more explicitly. And if you want to have a practice with the tools that you already know, how to have like an internal representation of the cell cycle, please reach out and we can do that. Like it's gonna take like half hour. I promise. It's not very complicated. But what we were doing before though was that we were the way we were representing the cell cycle was by having our cells grow, by having the volume of our cell increase, and then by having our cells divide when it reaches a specific size. And that's just that is a video of how cells do it actually. And I think that's an embryo. Yeah, I don't know where okay, so the first thing that we talked about last time is that we can make our cells grow by increasing its target volume. And note that it's important to know, because this comes up in the homework, that this step function inside every CompuCell steppable is called every Monte Carlo step, right? It's called every simulation step. So the growth rate of the cell is gonna be determined, right? It's gonna be whatever number I increase the cell volume each simulation step, right? Per simulation step. So in this case, my cell is growing 1 voxel every Monte Carlo step. If I change that to 10, that would mean that my target volume is increasing 10 Monte Carlo steps, 10 pixels every Monte Carlo steps. In the homework, when we asked you to double the target volume over 1,000 Monte Carlo steps, right? What we were asking was, so if the original cell target volume was 30, what we wanted is to increase the target volume 30 pixels, right? Because that's doubling over 1,000 Monte Carlo steps. So you would have something that looks like 30 over 1,000 instead of 1. And we in this case, we have unlimited growth, but we'll get to the point where this growth is gonna be constrained by something else. We'll see that a little bit later in the class today. So once we have our cells grow, then we can have our cells divide as well. And the way we do that, we explain that we have a special steppable in CompuCell that is called the mitosis steppable base, that it needs to be is the steppable in which in which cell division is executed. So you cannot do cell division outside of this mitosis steppable. And this mitosis steppable has the same structure as the other steppables, right? It has a step function. For example, there is nothing that prevents you from having your cell growth being specified in your mitosis steppable. You could do it there, but you cannot do the other way. You cannot then have your cell division happen in your, let's say, your growth mitosis, in your growth steppable. So just keeping that in mind, just in case you run into problems, either your cell division is not happening or your simulation is crashing, make sure that if you are calling the cell division methods, that they are being called inside the specific steppable they need to. And here we, as a condition of for cell division, we said that once the cell volume, the actual cell volume of the cell becomes larger than 50, then that cell is appended to this dictionary called cells to divide. And then, let's see. Yeah. And then once it's been appended, we iterate over that list of cells that to divide, and decide what are the orientation of the cell division plane, right? And we did explore a little bit of this. The cell can have a random division plane. It can have a specific direction in which it divides, or it can divide along its major or minor axis. And by that, we mean, let's say a cell is elongated, the major axis will be the axis in which the cell is the longest, and the minor axis will be the in which the cell is the shortest. And those have they might seem arbitrary, but they are actually important, especially in developmental processes. We were working at some time in a simulation of James, what's that animal that grows like a tube when it's dividing? It's at the sea. It's like one of like a sea sponge that when it's dividing, it divides. When it's in development, it grows like a cylinder, and that is mostly regulated by the plane of the cell division. So it is important in terms in certain biological processes. The division of that plane is important. The orientation of that plane is important. And then we have the mitosis stable also has this additional. Do you guys see my cursor? Yeah, okay. Has this special function called update attributes, and that function is called on every cell, and it decides what happens to the cell once it divides with its properties, right? Just as we were talking, the cell has a cell volume, has a cell or a target volume, a target surface, and what this update attribute function does is it determines what happens to those properties once a cell divides. What we usually do is that once a cell divides, we update its target volume such that it's cut in half, right? Because we want the cell to divide evenly. If we wanted the cell to divide asymmetrically, then we could play with what the target volume of the parent cell would be as opposed to the target cell of the daughter cell. If we just want to clone all of the parent's property to the child, we just have this function that also takes up the property of the mother cell or the parent cell and clones them to the daughter cell. And then at the end of the class, we looked at what happens in our simulation if we have cell division occurring in combination with constant cell growth. And what we saw is that the cells eventually filled up the simulation space, and that the pressure associated with each cell blows up, right? And that is due because cells don't have any more space to fill up, but the target volume keeps increasing in time, right? So what we're gonna, no, I'm sorry, no. One way around that problem that we were working with last time was to have two different types of cell, right? This is something that you actually see in stem cell proliferation, right? Where you have one cell that is the cell that it can actually divide, and then one cell that is what's called terminally differentiated, where they don't actually divide and create more progeny. So we had one cell that was the proliferative cell type, and that one would divide, but the cell type of the daughter would be different, which would be the quiescent or also terminally differentiated, and that one wouldn't divide. So what we will see at the end of the simulation is one cell in the middle, which is the green one, which is the proliferative one, basically surrounded by all its daughter cells, which were terminally differentiated. And that's something we see in, for example, wound healing. And we also see it in I think, James, it's seen in the replacement of the lining of the gut, right? And in the colon crypts as well. So this is actually something that And depending on of the interactions between cell types, and they can actually adopt very complex morphologies. So now we'll come back to this problem, but we'll address it in a slightly different way. What we're gonna do is we're gonna try to represent what it's called contact inhibited growth. In that case, the cells stop, because in reality, especially if you look at wound healing, what happens is that cells, once you get a wound basically disrupts the morphology of the tissue, it creates a hole. Cells detect that hole, especially the stem cells, and they start proliferating until that hole is filled up, right? And then the way they know how to stop differentiating is because the space has been filled up. So there has to be a way of cells to know that they are in contact with other cells. So for example, there's a very nice images I probably added to the slide deck of they marked cells at the edges of the wound to see which cells are proliferating and which are not. And you can see that the edges that are the cells that are surrounded by other cells are usually not proliferating. And then the ones that are at the edge that have some free space, those are the ones that actually proliferate until they fill up and fill up the wound. So we can do that in very different. There's different ways of doing it in CompuCell. What we will do as a proxy to that is to make the growth rate of our cells be dependent on the pressure. And as we mentioned, the pressure is the difference, right? It's determined by the difference between the cell volume and the cell target volume. And what you will see is that if the space is filled up and there's no more space for cells to grow, that cell pressure is going to increase. The pressure is going to increase. That's what we saw in the previous simulation. So what we want to actually do is to have our cells, the growth rate decrease if the pressure of the cell is increasing. Right. So the way we will do this is as described in, this is just a repetition of what I said. We will use pressure as a surrogate of the contact inhibition. But what we'll do is that we will increase the target volume, but make that increase a function of the pressure of the cell. Right, and we can make it a function of the average pressure in the simulation domain, or we can also make it a function of the pressure experienced by the cell itself. And there's multiple functional forms that relation can have between the pressure and the growth rate. But in all of them, what you want it to be is a decreasing function of pressure, right? As this pressure increases, you want your growth rate to go down. And what we will use a form that looks, the problem with using a form that is linearly decreasing, like this one, is that at some point, then the growth rate becomes negative. And we do not want that. We will have to have like a discontinuous function where it decreases linearly until it gets to 0 and then it stays at 0. Or we can have a more continuous function where it's smoother, the transition is smoother. And how we can play with this function to make it almost kind of like a switch, where cells go from growing and then they stop growing. And we'll play with that in the exercise. Okay, so this brings us to the first in-class exercise. I'm gonna stop sharing so I can share my full desktop. Do you guys see my desktop now? My full screen?
Yeah, we see it.
Okay, so the first exercise is we're gonna create a simulation where the cells grow and divide. And the first thing we're gonna do, and takes us back to the homework, is plot the average pressure over the cell. So we're gonna create a simulation and tweet it. Let's call it 6.1. Oh, okay. 6_1. No, what? One second. Here.
It may have logged you out. You may have to log in again.
Yeah, have you had this problem before?
Yes.
Okay.
Sometimes your credentials stale and you have to log in again if you haven't.
Okay. Let me refresh this. No, let me terminate. Move this out of the way.
I don't quite understand. Sometimes it seems to keep you logged in and active for hours or days, and sometimes it knocks you out quickly.
Yeah. I'm definitely not gonna be able to write in that one.
User, you have to find your home.
Yeah, I'm not exactly sure.
User.
User. Let's go here. Here. So supposed to be home. Home. Nano hub. And that's
There you are.
okay. There we go. We can keep it in 256 by 256. And we will use a Blob Initializer. And we can play with this whether we want it periodic or no flux. It won't make a difference for the moment. Let's initialize a cell type. I'm just calling it cell. Not gonna need this. We're gonna need the volume local flex, and let's just specify contact so they stick to each other. And that's yep.
Not growth or mitosis.
I'm sorry. I'll add them. So if that happens to you, what I'll do is, as I mentioned before, you can specify the cell growth inside of other step hours. The advantage of checking the growth step hour is that it already provides you with the snippet codes that you're gonna need to to make your cells grow. But if you forget that, then you can have your cells grow on any step level that you want. But what you do need is the mitosis step level that you're gonna need. So to add, as let's say you forgot a step level, you can right click on your Python step level files, and you can click on add step level. You're gonna have this window, and then you have different types of step levels that you can pick. The genetic one is just a basic step level. Mitosis is the one that you need to actually implement the mitosis. The other ones will come back to them later. But let's just choose a mitosis step level now. And it automatically adds a mitosis step level, and I keep hitting Command S to save. Automatically it adds it automatically adds the mitosis step level to your Python file, and it also adds it to your registry of step levels. So you need to save both files. By the end of the class today, I'll know not to hit Command S. It's just automatic. Okay, so let's come to the XML file. We want to initialize one single cell at the center of the simulation. The center of the simulation is specified there. Let's say that the radius is going to be 3 and the width is going to be 5. The volume plugin is loaded, but it's not specifying XML, which means that I can specify it, or I have to specify it in Python. So what we want to do is that we want first to have our cell grow and then divide when it reaches our double its target volume. So I iterate over, since I haven't specified it yet, right? I have to specify it in CompuCell. The first thing I'll do is, I was hoping it would have once you click the local volume, doesn't it give you like a basic snippet of the target volume on the cell in Python?
I thought it did. I think if you put the growth steppable in, it does.
Oh yeah, absolutely. So that's the problem. My problem is that I didn't put the growth steppable, right? So my cell does, as we said before, we don't get anything for free. So when the simulation is initialized, I haven't specified what the cell volume is. So to do that, I go to the inside the start function and I iterate over my list of cells, and then I assign a target volume, let's do it 25, and a lambda volume. Okay. And then now I want to make up my cell growth, right? And what I'll do is I iterate over my cells, and I will increase my target volume. Let's call it growth rate here. Let's say I want my cell to increase 50 pixels over 500 Monte Carlo steps. Right? So that is the growth rate of my cell. And now what's going to happen is that my cell is going to grow and it's going to divide. But I also want to plot the average pressure of my cells. To do that, I can use the tools available in the pull down menu, CC3D Python. And I actually don't know this by memory, so I always use this because it's a lot of code. I go to scientific plots and I set up my window. Notice that the indentation is incorrect. And we're gonna call it average pressure. So people were asking in the homework how to turn on the grid if I wanted to. And in the previous lecture, I also had some snippet of code that allows you to turn on what the legend of the plot is. I just don't know it by hand right now, but I can get it a little bit later. We're gonna call this average pressure, and let's make it a line because it's just gonna be one, right? Well, by convention, what I prefer to do is if it's gonna be something that I iterate over individual cells, I do points, and then if I wanna do something that I know it's gonna be it's gonna be one data point per time, then or per x corresponding to the x-axis, then I do a line. So I have initialized my average pressure. All I need to do is to calculate the average pressure per cell. So what I'm gonna do is I'm gonna define this variable and initialize it to be 0. And then I'm gonna, or let's be more specific. The total volumes of my cells. And then as I iterate over the list over my cell list, which contains all of my cells, then I can add each independent cell volume to this variable.
I don't think you want to do that. I think you really want to add the pressure at each time.
Oh, I'm sorry. Yeah.
Although you could do it the other way. It doesn't
Right,
matter.
right. No, it's fine. I'm sorry. I was thinking about
it's
plotting
linear.
the average cell
The
volume.
pressure is linear, so it's okay to do it the other way.
Right, right. But it's fine. We'll do it. What we want to do is not the total, is the total pressure, right? And the total pressure. And that is gonna be -2 times cell that target volume minus cell volume and lambda volume. And once I'm outside of this loop over
You
my
need a
cell,
plus equals.
once I'm outside of this loop where I have added all the pressures of my cell, then I can I can define my average pressure to be my total pressure over the length of my cell list, which contains the number of cells that I've iterated over, right? The length of the list of my cells is just how many cells I have. And then I can add my data point. And notice that this is just going to add one data point per Monte Carlo step, right? Because I'm outside of my cell loop. It has to have the same name. And what I want is my average. So that is going to plot the average pressure of my cells. So I have defined a growth rate, and I've plotted the average pressure of my cells. And notice that I have my cells divide once their actual volume reaches reaches 50, which is I'm sorry, has to be 25. When it reaches 50, which is the double of their initial size. And they divide on a random orientation. And once they divide, I cut their target volume in half. And since I have just one cell type, I don't need this snippet, which is just changing the cell type of the daughter cells. Okay, let's run the simulation now. It crashed. Oh no. So NanoHub is an excellent educational tool. But for example, now that it crashed, I have no way of checking whether I've made an error. I had an error in my code. Yeah, if it crashes, yeah, I definitely have an error somewhere. With your local versions, you would get like a segmentation error that will let you know whether you have something incorrect in your Python file or in your XML. Okay, let's open Twitter. And let's get the simulation again. Everything is fine here. I'm not sure. Let me. As yeah, this should work.
It must be crashing in the start because you never get one step.
Yeah. I'm not sure.
That
Just-
kind of crash usually is an XML problem, but you didn't handle, you didn't. you
I just
didn't-
changed this.
How did you change it?
I set the radius to 3 and the width
Be
to
all
5,
right.
but that's, yeah, that shouldn't be the problem.
Why don't you try launching it again from the Twitter? Don't go to player. Do it from Twitter.
It doesn't even let me from here.
Go put your cursor over the root. Put your cursor. Is it an issue of using a numerical name for the file?
I don't think so.
I've never tried that, so I don't know.
I think I've had issues using underscore before.
Yeah,
If you look at your steppables file, the class name is in red. And for me, whenever it's been in red, it means it's not working.
yeah, I wouldn't. Yeah, okay. Yeah, it's an illegal column. It's an illegal name.
Oh yeah, the slash. It might be the slash in the name that's
Yeah.
doing some weird stuff.
Okay.
I wouldn't ever use a numerical value for the name of anything.
Let's do something for the name of. Not even like versioning 1.
Not
Yeah,
at the
what-
beginning. Not the beginning of a name.
Yes, what's actually happening is you cannot start any variable or class name beginning with a number. It's fundamentally deal with the actual
Right. It's not just, it's just not right. So,
yeah. I mean, you can have a number in there later, but
right,
the parser itself
right.
cannot differentiate between that and a
Yeah,
number.
thank you. Thank you. Yeah. So here. So if you ever get a problem with that type of problem and you don't want to redo, let's say you have a very complex simulation that you don't want to start over, you could always rename the steppable classes, such that when you call them, yeah. Yeah, I need to rename this one. I mean, or yeah. you could always and I did this with someone in class the other day. You can rename your steppables, but I'll just start over really quick to avoid that problem. But thank you Andy, because yeah, that was clearly not a good choice. So do not start your projects with numbers. I'll just go really quick over what we did already. And this time I'll add the growth step up, the volume level
Selected
step,
uniform initializer.
and I'll add the growth stable and the mitosis stable from the very beginning. Okay, now, again, here, so now I don't have to specify the original volume because in the constraint initializer is specified. But I'll use this constraint initializer to initialize my plotting window. No, my growth stable, because it's the one where I'm the same one. So let's say I want to plot based on the information, I could always, if it's not specifying the stepable that I want to, I can always create a start function myself to inside whichever stepable I want to add any other information that I want to add to that stepable that might have not been specified. James, one question: when you use NanoHub, can you use the shortcuts, or do you have to do it like from within NanoHub as well?
Sorry, what do you mean?
So because I cannot use my command copy and my
It worked for me.
It works from you. So maybe people who what about people who have Macs?
I have an issue with the commands too.
Yeah, okay. Okay, that's good to know.
Odd
so,
that it's a Mac versus Windows.
growth rate, and then now I have my total pressure, and I want to add it.
One thing you could do maybe as a sort of a halfway thing is if you under edit, you have copy and paste. I don't know if those work.
Yeah, they do work, but I have to go like myself and copy and paste.
Right, but at least you don't have to retype everything.
Right. 2.0 times cell that lambda volume times cell target, that target volume minus cell then volume, and then now my average pressure is gonna be my total pressure divided by the length of my cell list. And then I add my data point. Gonna be okay. And then again, remember that we don't have a second cell type, so we can get rid of this part where we change. Maybe that code snippet should be suggested instead of being automatic. And last thing is we want one single cell at the center of the simulation domain. Okay, that should work. Get this out of the way. That's not the one. And there you go. So now we let it grow. I wanted to make it right. Not sure what it's.
You didn't define a surface energy, contact energy.
I didn't. Okay, so if you don't define contact energies, all of this stuff can be loaded afterwards, right? So we come to the XML, we go to plugins, and we can specify contact. So let's say you forgot to do it, just like as I did. I forget to do it in my initialization. I can always come back to this XML file, and using the CC3D pull-down menu, I can specify what my contact. I can load the another crash. I can load my my plugin, and I can using the pull down menu. Just again, just as just for you not. So you need, if you, let's say, forgot to do something, you can come back and do it later. Everything that is available to you in the initialization window will be available to you as well in the in the XML. Not sure. Not sure why it crashed.
That looks okay.
Yeah. So now you can see the cell increasing and the pressure the pressure stays relatively constant at the beginning because we have plenty of space, right? If we were, let's speed this up by increasing the growth rate, right? Where we define the growth rate we define the growth rate to be 25 pixels over 500 Monte Carlo steps. Let's do over 100 Monte Carlo steps. So we're speeding it up 5 times. I'm gonna launch this. Okay, so we'll see the same. The pressure should stay relatively constant. The average pressure. Why it's not going up? Okay, let's come back to the code.
We left out the plus and the plus equals.
Okay, now. Yeah, there you go. So now, as the space gets occupied by other cells, and also as the cells in the middle have less space to grow, the pressure is supposed to increase, right? So now what we want to do is we want to measure the pressure in the center of the simulation domain. This is consistent with what we've seen so far, the pressure blowing up because the cells occupies the space, right? The first thing we're gonna do now is that we want to be able to plot the pressure along, let's say, a cross-section of my simulation domain. So to do that, because my pressure is a property of the cells, it's not a property of the pixels, right? So what I'll do conceptually is that I'm gonna iterate over each, let's say each pixel in a line in my simulation domain. I'm gonna identify what is the cell that occupies that pixel, and then I will plot what the corresponding pressure of that cell is. So we're gonna need another plotting window. And we're gonna call this one, pressure per location or something. Notice that now my x-axis is not gonna be time anymore, but it's gonna be my location in X. And my y-axis is gonna be the pressure. Now I need a different name for this, and we're gonna call it, we're just gonna call it, we can call it pressure. As long as they are different, it's fine. Okay, so now what I need to do is, as I said, I need to iterate through my simulation domain. So let's call each location in my simulation domain along this line X. So what I need is to iterate from 0, because that's my first location, to the last location in that around across that dimension. And that is when you call this function solve that then that x, that gives you basically the size of your simulation domain in the x dimension. So I'm basically iterating from 1 to 256. Now I need to visit that cell that is at the specific location in X. So there's a convenience function in CC3D Python pull-down menu where I can
It's under a cell attribute.
write cell attributes. Is
It's cell manipulation there, yeah. Cell manipulation.
Yep.
Get cell add a pixel.
Get cell add a pixel, right? So this is gonna tell me what the cell is at the specific location in the X, Y, and Z, right? So X is the one I'm iterating to. Since I want it at the middle of my simulation in the Y dimension, what I can do is use this same property and divide it by 2. And I think 0 is fine because 0 is the first domain in the Z-axis. We don't have any other. It's the first location on the Z-axis. We don't have any more. So now we're picking that particular cell. And then what I want to do now is that I want to calculate the pressure of that cell. And I want to see that using the cell field, I define this object called cell that is at this particular location. And now I can calculate, now that I have identified the cell, I can calculate the pressure associated with that cell. And then I can add a data point.
You may want to get rid of the minus sign in your pressure just so that you have positive pressures.
Yeah. And now I can add the pressure associated with that cell. And notice that in this function at data point, I specify the data set that corresponds that I'm adding the point to, what I'm adding on the x-axis, and what I'm adding on the y-axis. And question for you, what should be in here? Because when I plotted it as MCS, I'm plotting as a function of time. But this time, that's not what I want to plot. What I want to plot as a function of what? I want to plot my pressure as a function of what specifically?
Is it of X location?
Absolutely, yeah. So it's gonna be, my plot is supposed to be X location and the Y-axis of pressure. So yes, it's gonna be the particular X location and the pressure.
It's just going to crash because your plot window's got the same handle.
Right. Good point. So handles need to be different, right? So I'm already instantiating the plot window using this handle. So I need a different handle for my second window. So now I have two plot windows instantiated, one instantiated with this handle and has this data set associated with it. A second plot window with this handle and another data set. And I am adding different types of points to them. What?
You have to do an if cell.
Mhm. Right, because if there's no cells in it, if there's no cells, if it's medium, medium does not have a lambda volume associated with them. And if there's no lambda volume and there's no actual volume, then the pressure should be 0. So it's 0 unless there is a cell occupying that space. And when there is a cell occupying that space, then it's defined by the difference between lambda and target volume.
Indent.
No, where?
Line 46.
Oh, there's a right there. There you go. I don't know what's happening with my CompuCell. Do you guys have the same type of problems with CompuCell? Okay, so now you guys can see that as the average pressure is increasing, and also the pressure by location is increasing. Let's do something. Let's plot. And this is also important when you guys do it and you see, okay, this plot doesn't seem to be very informative. You guys can change the properties of the plot to see whether other properties might be more or less informative.
Try resizing player so it fills the window correctly.
Why is player that weirdly?
I don't know. It's
That you guys see. So you guys can see how the pressure is changing by location, right? As my cells grow and divide. As they grow, as they grow, you'll see that pressure at the center increasing as the average pressure increases as well. So what you should see is an increase in the average pressure. Also, I've noticed, James, that we didn't mention this in the homework, but this plot is not refreshing, so it's plotting on top of each other. So if you want to refresh the plot, so every time you'll see a different plot of the pressure. So because the reason you're seeing all these points here, it's because each Monte Carlo step, I am plotting more and more data, but I am not refreshing the plot window. Whereas in reality, what I want to see is the plot refresh every time that a Monte Carlo step is called. If I come to CC3D Python scientific plots, I can erase plot, erase all data at the beginning of the step or before adding. Before adding my new data. Do you guys see what I mean? In this plot here, I'm basically adding all of the points on on top of each other. What I really want to see is every time I plot the data, plot the pressure as a function of the location, I want a new plot. And that I do by calling this erase all data function before plotting the next step. And also, as you guys can see, this is quite computationally expensive. Iterating over pixels is usually computationally expensive. That's why the simulation is taking a decent amount of time. But I'm gonna move on so we can get to the fields. But let's talk about this last part, right? In this part, what we want is to make that our growth rate a function of the pressure. And we saw already that the pressure is increasing exponentially as the cell domain gets filled up. What I would do to account for pressure inhibited growth is once I have to make my growth rate a function of my cell pressure, right? So I've calculated the cell pressure here. Let's cut this. And now I want to make my growth rate a function of my cell pressure. So what I'm gonna do is I'm gonna comment out this for now so the simulation runs faster. Okay, so now I want to make my growth rate a function, as we said, a decreasing function of my pressure. So what I will do is that I will define a max pressure that I'll let my cells experience. What that max pressure is, what a reasonable max pressure is, you can either determine by looking at the cells and what is a reasonable pressure that keeps them having, and I was talking to someone earlier about this, that makes them have a particular shape and form and motility. That was part of the homework. You can also infer that a little bit from the equation itself. We'll just set it to be such that this difference is the max pressure, such that the difference between is proportional to the difference between my target volume and actual volume. And let's say is let's call it top 5.
I don't think you want to do that. I think you want it just to be a number, like
Okay.
20.
Let's just set it to 20. So now what's going to happen is that if my salt pressure is much, much bigger than my max pressure, then my growth rate is going to go to 0, right? Look at this equation here. Let's say salt pressure is very big. Then in that case, this part turns into 0, which means that my target volume will stop increasing because of the pressure.
There's one other thing you have to do, which is you have to make sure that you max the pressure with 0, because you don't want the pressure to be negative.
Right, but since this is a decreasing function, what I'm decreasing is not the cell target volume, right? I'm decreasing the growth rate.
No. But still, your cell pressure could early on be negative.
Oh
because
yeah,
of
that's true.
pressure. So you need to max the cell pressure with 0 to make sure it doesn't.
Right. So
No, you have to do it when you calculate the pressure. Because if you make, you have to do it in line
the max pressure
38.
and the cell volume, cell pressure. I'm just trying to identify what the typo is because there is a typo somewhere. Okay. Here.
Line 38, you need to make that, max that with 0.
And I'm not sure.
Is that throwing an error?
Yeah, it's saying that there is a syntax error, but I'm. Okay, so yeah, that's basically making our growth rate being dependent on the salt pressure. And what will happen is that as cells start experiencing more and more pressure, then they stop growing. And you can sort of like see it quickly because in our previous simulation, the average pressure was increasing very, very rapidly. Whereas here, our pressure sort of like increases much slower. And let's see what happens when the whole simulation domain fills up. And how much of this you get, how much is going to be determined by, of course, what is the maximum pressure that you're allowing in the simulation, right? But this is just an example of how you can basically control how much of how the growth rate is going to be determined by the growth rate of the cells is determined by the pressure they are experiencing. And you can start seeing here that that already the pressure is starting to saturate instead of growing exponentially, which is what we wanted. Okay, but I'm gonna stop here and I'm gonna keep going because we wanna cover other topics. But if you guys have more questions about this, I taught you how to iterate over the simulation domain and then how to make your slope growth dependent on the pressure. Okay. So, okay, let's talk now about fields. In cell biology, chemicals are, when we talked about chemicals in the context of tissues and cell, we usually we refer to molecules that are discrete and occupy certain spaces, and they come into contact with each other, and they sort of like are discrete entities such as cells, right? But when we looked at, let's say we looked at molecules and chemicals from the perspective of tissues, we usually look at, we are looking at them not just as individual cells, but or individual molecules, but as collections of them. And at this when we talked about this big collection of molecules, then we can stop treating them as single entities and treat them as densities, as basically as we can, instead of treating them each individually, we can talk about how many of them occupy a specific volume of space. And that is what we called a concentration field, right? We start looking at the chemicals as individual molecules, and we treat them as concentrations. We give a number that specifies how much of these molecules occupy a specific amount of space. And again, when do we treat one versus the other? When we treat molecules as individual entities versus as concentrations, it's going to be dependent on the specific problem that you might be interested in. But in general, when we talked about in tissues, you can confidently treat molecules as fields, because in this particular type of context, we are referred to collections, big collections of molecules. So the idea here is that instead of thinking about individual molecules, we're gonna think about chemicals as concentrations, right? And these concentrations, the most important thing about concentrations is that there are differences in concentrations across tissues, across space, and that there are mechanisms that ensure that these concentrations are changed over time. Some of these mechanisms, and here's a list of a couple of the mechanisms that change concentrations, that enforce changes in concentration. Some concentrations change because of advection. Geoffrey was talking a little bit about that earlier. Some of them change because of active transport, and importantly, in the context in which we usually work, then diffusion is an important mechanism for transport and changes in concentrations, and we'll talk about that more in detail. Advection refers to the movement of chemicals in a, let's say, because of a particular flow, because let's say a cell has a particular concentration inside of it, and it moves and it carries the concentration that it has inside with them. We have certain limitations in simulating advection in CompuCell. So we usually have to do those changes in concentration due to advection. We usually have to implement manually instead of having CompuCell solve those changes for us. There are other mechanisms in cell biology that ensure changes in concentration, and active transport is an important one. Cells have this transmembrane proteins that ensure that are ensure that concentrations across membranes are equilibrated or maintained. And they do so by taking molecules from the outside of the cell and pumping it in, or by taking cells from the inside of the cell and pumping them out. That it's known as active transport because it actually requires consumption of energy by the cell to maintain these differences in concentration across their membranes. In cells, they also get some type of vesicular transport where certain chemicals are put in certain vesicles, and these vesicles are transported inside or outside of the cell. And that's how you get, for example, endocytosis inside the cell, and you get exocytosis outside of the cell. You get a transport of neurotransmitters that way. That's interesting video of the molecule walking. I mean, there's multiple of them. Actin is one of them, but you can also get transport of neurotransmitters that way. But the most important one that we're gonna be focusing on is diffusion. And in diffusion, transport occurs merely because of different in concentrations and random motion of the molecules. So let's say you drop a drop of ink in liquid. You don't have to move, let's say, your glass to make that drop eventually mix up in the whole liquid. Although it takes time, eventually, because of the random motion of the molecules of ink and the collisions with the molecules of water, you would get diffusion of ink all over the liquid. So in general, diffusion occurs because of the changes, because of random motion and due to this differences in concentration. And what you expect to get from diffusion processes is that molecules will move naturally without energy, without the consumption of energy, from areas of high concentration to areas of low concentration. And that would eventually erase the concentration gradients. And by concentration gradients, we mean the difference in concentration over a domain. Because diffusion is due to these molecular random motions, they take a lot of time, and it's mostly important over short distances. So cells take advantage of diffusion to have chemicals move from one point to another over short distances because they don't have to use energy. But if, let's say, an axon needs to transport something from an axon, a neuron that is very long needs to transport some chemical from across the axon, that will probably require some type of active transport, because the cell would not be able to rely on diffusion to get the chemical transported in a reasonable amount of time. So again, diffusion is more important over short distances, usually like up to a few millimeters. As I said, the distance and then that distance, and the reason is because the distance, if you calculate the diffusion, if you based on the diffusion equation, if you calculate how long it takes to move a molecule to travels, that distance is going to be proportional to the square root of time. So if a molecule travels 1 second, it travels 1 micrometer in 1 second, then it takes 2 micrometers, 4 seconds to travel 2 micrometers, 100 seconds to travel 10 micrometers, and so on. So it increases squarely, and it takes more and more time as you move more and more distance. right? But cells do take advantage of this random movement because they do not have to, as I said, they don't have to exert energy. And it's just very cheap and efficient for them to take advantage of diffusion over short distances. So let's say we define now let's look at mathematically how to define how to think of diffusion mathematically. And we start by defining a concentration, let's say on a specific voxel or a specific pixel or a specific lattice at a particular location and time. Then the flux, right? How much of the material is coming in or out of that voxel is going to be a function of the difference in concentration, right? How much you have, let's say, at one edge of the flux due to diffusion, right? How much you have in one side of the one edge of the voxel versus the difference at the other edge of the voxel. If you want to know how the concentration is going to change over time inside that voxel, then what you need is not the flux, but the change in flux. And that is what you get with the diffusion equation. The diffusion equation describes how the concentration in a particular location changes as a function of time due to the gradient, which is what we call the change in the flux, or if you want to look at it another way, the second derivative of the concentration as a function of their location. And we'll see that later on as well when we plot the concentrations as a function of location. But this is important because this determines how the concentration is going to change at a particular location as a function of time. And notice that there is this parameter here, D. That parameter is what we call the diffusion coefficient. And that diffusion coefficient sets the time scale at which diffusion is occurring, right? So diffusion occurs because of the change in concentration, because of the gradient, and at a speed that is proportional to that diffusion coefficient. Notice that the diffusion coefficient, so we talked about one particular location, right? Now let's think about like a whole tissue patch, right? The diffusion coefficient might not be the same at every particular location of the tissue because a molecule might diffuse easier, let's say in water, than inside the cytoplasm of a cell. And why would that be the case? Any ideas why a molecule might diffuse faster in water than inside a cell? Let's say I have, I don't know, a molecule of glucose. Glucose is not going to diffuse through the cell, but let's say you have like a molecule of water diffusing water, or a molecule of oxygen diffusing water. Why would it take longer for it to diffuse inside a cell than to diffuse in water? From a cell biology perspective. Any ideas? So the reason, like from a biological perspective, is because the interior of the cells are usually very compact. It's usually full of stuff. So it's harder for a molecule to move from let's say one side to the other one. Whereas in water, they just have to move through the water molecules. But usually the inside of the cell is much packed, much denser than the outside. or the interstitial fluid. Okay. So yeah, that's the idea here. Let's say if you have a chemical diffusing over, let's say, water or a free media, they will diffuse much faster than if they are diffusing over a medium that is obstructed by either other molecules or by impermeable membranes. Okay, and we will play with that one, with differences in diffusion across tissues in an exercise later today. So how do CompuCell represents chemical fields? It's quite simple. The idea is that we define an object called a field, and we can name the field with whatever name we want to represent the chemical that we of interest. And then CompuCell is going to associate a number at each pixel that represents the concentration of that molecule at that particular location, at that particular pixel. And then based on then CompuCell will invoke partial differential equation solvers that are responsible for solving the equation, the diffusion equation for that, over the simulation domain, right? And again, the purpose of because of diffusion, the net effect of solving those equations is to flatten the concentration differences across the simulation domain. And we'll play. That's the purpose of the next exercise. CompuCell has a couple of different of these solvers. The details are not very important. For now, we will use the diffusion solver FE, but based on the conditions that you want to represent, there are other diffusion solvers available to you that you could use.
Yeah, I think so.
Okay, so CompuCell, using Wizard, we can create convenient. We can use the Wizard to define what the chemical fields will be and what the diffusion solver associated with these chemicals are. I'll walk through this really quick because we'll do it again. When the diffusion chemical fields pop-up window pops up, you can define your field name. Then you choose what are the diffusion solver that is going to be associated with that field. And then you add it just the same way we add cells. You hit add to add that field, and then you can click next, and then that adds the field to the simulation domain. That's going to create this diffusion solver steppable in your XML file, and that XML is going to contain the details of the diffusion of the diffusion solver that you're going to use. And as we mentioned, the most important parameter that we're going to have to specify is this diffusion constant, which is going to tell how fast the molecule diffuses across the simulation, right? And for now, we're going to define this diffusion constant globally, although we can also define that we can define it differently under other circumstances. We can also define what the initial concentration of our field will be. If you leave that commented out, the solver assumes that there is initially no amount of field, no concentration in the simulation domain. You could also import the initial conditions of the chemical concentration as a text file, right? All you need is to specify what the value of the concentration is per simulation domain. right? So again, because the idea is that CompuCell represents concentrations as these numbers associated with every lattice. The last thing you need to specify the concentration, the diffusion solver is the boundary conditions. And as we said, boundary conditions specify what happens to the chemical field at the edges of the simulation. And there's a couple of stuff that could happen to the chemical field. The first one is what is known as periodic boundary conditions. And this is the illustration here. So periodic boundary conditions assume that the simulation, the domain that I'm simulating is just a smaller section, a smaller fraction of a bigger simulation, of a bigger domain that is basically mirror copies of the domain I'm looking at. So what happens is, let's say this particle moves across the edge on the left side. Since there is a mirror copy of that domain on the right side, that molecule will reappear or re-enter through the right side, right? So it will disappear through the left side and will appear to the right side. Another way of thinking about it is as the simulation domain was wrapped on itself, so as if it was circular. So that is what periodic boundary conditions are. So again, if it moves up, it appears on the bottom. If it goes right, it appears on the left side. Constant flux determines that at the edges, there is the concentration changes every step by a single amount, right? So let's say I have a constant flux that is negative in this edge, on the right edge. It means that concentration, let's say it's -1, every step, the concentration at that edge is decreased by -1, right? At the edge, at the boundary. And then the last option is that there is a constant value at the edge. Let's say I specify that the constant value at my edge is going to be 1. Every step, the concentration at that boundary is always 1. And then diffusion works according to what happens inside of the simulation domain. It's got to respect the fact that at the edge, the value is always going to be 1.
You might mention that constant flux of 0, no flux, is basically building a rigid wall that doesn't let molecules leave.
Right.
And constant value of 0 is absorbing boundary conditions, where anything that touches the boundary disappears.
Yeah. Thank you, James. Right. If I set my constant flux to 0, that's basically saying that I put a wall, and if a molecule comes this way, it has to move back, right? So because it cannot go through my edge. And if I set the constant value to 0, it means that always at the edge there is value 0. So basically the molecule goes and disappear over my edge. And we'll play with that for sure in the exercise. But when you load the diffusion solver, besides the diffusion parameters, you also have to specify what happens at the boundary, and it's also loaded in the XML. So you can, as we said, you have the choice to, you can choose periodic boundary conditions, you can choose a constant derivative or a constant flux, and a constant value. Okay. So okay, let's work on our first exercise. We're gonna create a simulation domain with one field and no decay. We'll come back to decay later. What we'll do at the beginning is that we will initialize our simulation domain with a concentration that is uniform across the tissue, right? And periodic boundary conditions. So I mentioned that because with diffusion, the net effect of diffusion is to erase changes in gradients in concentration, right? So what would you expect to happen if I initialize my concentration with a uniform with every value of the field being 1 at every location in the simulation domain? Basically, I start with a uniform concentration across my tissue. What do you think would happen? So if there's no changes in concentration, right? See, there's no differences in concentration, there cannot be changes in concentration. So I shouldn't diffusion, nothing would, there wouldn't be a net diffusion in my field. And we'll see that, we'll work on it right now. So let's create a new simulation. Oh, I'm sorry, you guys are not seeing my screen. Okay. Exercise 2, we need to save it in my directory. Let's do 200 and 200. So we're gonna have a 200 by 200 simulation domain. We said this is the boundary conditions for, these are for the cell lattice, and these are different from for the field. That this is important because I can have different boundary conditions for what happened to my cells versus what happened to my fields. For now, we'll leave this to be no flux. It doesn't matter because we're not gonna use cells for now. So as I said, we're not gonna initialize any cells, so we can skip this. And now we have the chemical field pop-up window, and let's define one chemical field. Let's call it oxygen. And we're gonna use the diffusion solver FE. But if you guys wanted to see what other options are for you as diffusion solvers, you can choose that. You can click on the menu, and you'll see what other options are. But for now, let's just use the diffusion solver FE and add the chemical field. We don't need any of this because these are sort properties and behaviors, and we're not using any cells at the moment. Okay. So if we come to the XML, we can see that the diffusion solver FE steppable is loaded, and that it already specifies a field called oxygen that I initialized. And it has a global diffusion constant, right? This corresponds to the diffusion coefficient that we talked about. Let's set it to 1. And no decay means that this line 38, we can comment out or we can delete. I'm not gonna delete it, yeah. Undo. Just to avoid any errors, I'm just gonna set it to 0. Okay. And then we said that we want the initial concentration to be uniform, right? On the field. So we can use this initial concentration expression. That allows me to specify what the initial concentration of the field is. And notice that I can use X and Y referring to my simulation domains locations to formulate what my concentration would be. For now, I want it to be uniform. So I will set it to be 1, which means that it's going to be 1 all over my simulation domain. We don't need any secretion. We'll come back to this later, so we don't need it at the moment. And now we come to the boundary conditions that we were talking about earlier. So we can see that we see the constant value, the constant derivative, and the periodic. And we will set it to be periodic for now. Okay. Now I can open my simulation. Oh, I'm sorry. I have the blob initializer. The uniform initializer. I don't want it. So let's just delete that for now. Okay. So since I haven't initialized any cells, I shouldn't see any cells in my simulation domain. And if I come to this pull down menu over here, I should be able to choose what I want to look at. I can look at my cell field, which is going to display the cells that are in my simulation, or I can choose to see what the concentration of my field is, right? And tell me what you guys see here. So you guys see that my concentration is the same color everywhere, right? And then when I look at this scale over here, it goes from 1 to 1, right? So the reason is because I initialize the field to be 1 everywhere, right? Let's run it to see what happens. So, yep. So what's happening right now? Any guesses? Because it's running, you guys see that it's running. But the concentration remains constant across my simulation domain. Why is that?
Since there's no like gradient stuff
Right.
diffuse.
Right. Since there's no gradient, because the concentration is uniform, there won't be any changes in my concentration, right? Because the diffusion is dependent on the gradient of the concentration, and there's no gradient, so there's no diffusion. Okay. Okay, so now let's try changing the boundary conditions to be constant derivative and constant value. Let's play first with constant value in the x-axis. And just as James mentioned, let's first make it 2. Min and max refer to, right? I'm looking at the x plane, and I'm looking at the minimum and the maximum of my x plane. And because those are the edges on the x dimension. And what I'm saying here is that the concentration at those edges will always be 2. Okay.
You want people to be following along and doing this while you're doing it?
Yes, James, can you pulled the poll. Yeah. So yeah, just to make sure that how many people have at least the simulation up and running. Okay, everyone's along. So now now that we have changed the boundary conditions, let's reset the simulation and see what we get. Okay, we see the same, but we know that it's not because we changed the initial conditions. And the problem is that well, I don't, did I save it? Yeah, I did. Sorry. Yeah,
It should be okay, just run it. Run it.
Yeah, but it's not setting anything. Oh, because I haven't come. I'm sorry. I need to comment this one out. Okay, now. Why is CompuCell taking so long here?
Try nudging the boundary of the.
There. Okay, so now let's restart it. Now you guys see that now is one everywhere except at the boundary, right? Where it had already started diffusing as soon as I hit step, right? But then when I hit play, what are you guys seeing? Can someone describe what they're seeing? Or explain what they're seeing?
We're seeing diffusion to the vertical, the Y boundaries on either side of our simulation space.
Right. So basically, the concentration is since it is fixed here, right, and fixed here. So there is higher concentration at the edges than in the rest of the simulation domain. So it diffuses in and it diffuses in, right? It never flattens out because the concentration is always inside the simulation domain. At the edges, I'm sorry.
If you run it long enough, it'll be two everywhere.
Right. It's true. Yeah, we'll see that in the exercise because there's no decay, right? But it's just gonna take a very, very long time. And that is part of the reason why diffusion might be not convenient over long periods of time or over long distances, depending on how you look at it. And then the last one that we're gonna do.
You can see the minimum value creeping up.
Right. So if you look at the minimum value,
is the minimum.
So
Here.
the minimum at the middle is gonna be at the middle, right? And it's increasing because basically this value is always 2. So the chemical is basically always sort of being, it's always coming into the sim. It's diffusing always from the boundary to the middle of the simulation. It just takes a very long time for the whole simulation domain for the chemical field in the whole simulation domain to reach 2 as a concentration everywhere. Let's do the constant derivative instead of constant value. So I'm now, instead of having a constant value of 2 always, but I'm always every time I'm basically providing, I'm sort of basically 2 units of my field are coming into my simulation domain from the edges. So I'm always adding 2.
Except you have to be careful about the sign. On the min side, you actually are taking it out.
Oh, really?
Because its direction is fixed.
Oh, yeah. So the flux is in one specific direction, right? It's not, I was thinking the flux is always like this, but the flux is always like this. So if you wanted it to be like this, you need it to be opposite signs at the 2 edges. There you go. So it's not necessarily radically different from what we saw before, but they do mean different things, right? They're doing different things in the sense that in one of the exercises, when I set the constant value, what I'm saying is my value at the edge is always this. Whereas when I set the constant derivative, I'm basically supplying every time 2 plus 2 plus 2 plus 2 plus 2 at the edge.
So you could see what the maximum concentration is here. Instead of 2, it's 100.
Right. Right. So, and that's another good point. When I set the constant value to 2, the highest the concentration can be in my field due to the concentration gradient is going to be 2. But here, since I'm always supplying in and I'm never taking out, the concentration is going to increase every step. The concentration inside of my domain is going to increase at every step. And it's going to continue increasing and increasing. So I'm going to leave 2, 1, 3.
The gradient will never disappear. It will
I'm sorry,
always,
go ahead, James.
in this case, the gradient will never disappear. It will always be less in the middle than the edge.
Right.
Whereas
Why is that the case? Yeah, that is another good point. James, question: what if the diffusion coefficient was very large or much larger?
Well, the larger the diffusion coefficient, the shallower the gradient.
The shallower that would be, right? The differential, the gradient will be, and we can play with that.
That you can do in the steering panel without restarting.
Yeah, I'm sorry, I restarted the simulation, but we can do. So let's use a steering panel to see what will happen if I change my diffusion coefficient.
Regarding the reason we don't see the vertical edges leave the maximum red value, well, it looks like that just keeps changing over time. So as long as there is more of the compound at the edges, it will always appear to have the highest concentration, unless maybe we let this go for a really, really long time.
James, do you want to explain that one?
So you're, in one case, if it's constant value, the system will equilibrate, because when everything has a value of 2, there's no more flow. In this case, you're continually pouring chemical in from the edges, and that chemical will gradually redistribute towards the middle. But you're always, no matter how big your value is at the edge, you're still pouring more in.
Got it.
And so in this case, because it takes time to equilibrate, you never equilibrate because it always is, there's always new material coming from the edge moving towards the center. Imagine you are making a cake and you're always pouring the material in from the edge of the pie pan. It's going to be a little higher on the edge than in the middle if you keep pouring. If you stop pouring, it will flatten out.
Right.
I could, let's see. Oh no, I can stop pouring in by setting, no, that's not gonna stop pouring.
Yes, it will. Take it, make the derivative 0 on the edges. That'll work.
Oh, right. Yeah. Because, right. So I basically stopped pouring in, but because it took me some time between changing one and the other, there was a change in concentration on one edge versus the other.
But you see it's flattening out. The difference between the max and the min, 317 to 304. And if you wait a few minutes, it'll be flat.
Right. And I can also, for example, increase the time. How long does it take for it to flatten out? But there is then the overcompensation of the bigger diffusion coefficient. Right. Or let's set it to 1, which makes it slower. So you barely see it's changing, but since I decrease the diffusion coefficient, now it takes much longer for it to equilibrate. Okay. So I'm gonna leave these 2 other exercises for the homework, and because we're gonna do more exploration about this. But the importance here is that we wanted to show what the effect of diffusion is, right? Is to erase the concentration gradients. And it's determined by the gradient of the chemical field. So if there's no chemical field, if there's no gradient, there is no diffusion. The bigger the gradient, the faster the change in concentration, etc. And then what happens when with different boundary conditions and how they change the gradient inside the simulation domain. Notice that I can change the values of my derivative, but I cannot change the type of boundary that I have. For that, I cannot do on the model editor. I would have to go back to the XML. So it's continuing on what we've done here with these differences in boundary conditions. Now we can talk about what happens if what happens with a, let's say, one-dimensional diffusion, which is what's happening here, right? It's diffusing along one dimension, not along the y dimension, because the concentration along the y dimension is the same everywhere. And we can solve under the assumption that there is no decay, and then that we have this fixed boundary condition, fixed values at the boundary. We can sort of solve the diffusion equation to determine how the concentration changes in time, and as a function of the location. And the important idea here is that if we fix the concentration at one edge, and we have a different edge, if we fix the concentration at the different 2 edges, the profile is going to be linearly it's going to be linear, right? It's going to be linear as we see here. And it's going to be independent of the at equilibrium, which means when the diffusion stops, what should I see is a gradient that is linear, and that the linearity of that gradient, the steepness of that gradient, is gonna be independent of my diffusion coefficient. How fast I approach that linear gradient is going to be dependent on D, but not the steepness of the gradient itself. And we'll play with that. So as to explain what we mean here. So what we want to do here is that we want to create a linear gradient between one edge and the other. So to do that, what we do is that we set the value at one edge to be higher than at as a constant value to be higher than at the other edge. Let's do it from scratch really quick. So what we want to be able to see is that linear gradient due to differences in the boundary conditions.
Why don't you just edit the one you have?
Yeah, I wanted to start it from the beginning, but yeah, we can do it here because we already
It'll be
have.
faster.
Right, yeah. So let's just set the value at the maximum to be 0 and the maximum, the value at the minimum of X to be 10.
You want value, not derivative.
Okay, so now that we have specified a constant value at one edge and a constant value of another one. And when we set that one to be 0, we set it, it meant that we set it as an absorbing boundary, which means that everything that goes through that boundary sort of like basically taken out. It basically, it's like it falls out, let's say, of a cliff. So what we should do, what we should see eventually is that this gradient is linearly distributed over the simulation domain. Let's do a couple of stuff so we can visualize this better. So one way we can visualize that is we can come to the configurations, and in fields would allow us to play with the visualization of the chemical fields. So for example, I could have the colors, in terms of the colors, I could fix what the maximum color is that I'm gonna be interested in, right? And what the minimum and the minimum of that value is. So I know the minimum of the field is 0 at the edge, and I know the maximum of the field is 10. So I can fix those so I get a better idea of what the gradient actually looks like. And then I can use contour plots, and the contour plots are gonna draw lines that corresponds to areas of the simulation domain that have the same amount of chemical. And let's say that we want 5 contour plots for now. Okay, so that changed, right? That changed the way my gradient looks like. And see how my line is still moving on this direction. And when we say that it's linearly, the gradient is linearly distributed across the simulation domain, we also sort of mean that because the contour plots are equally spaced, they should be equally spaced in the simulation domain when my plot reaches equilibrium, which it haven't yet because the contour plots are still moving. Is that right, James?
Yes, I think you started with an initial value of 1 in your field, so you might want to start with 0 value.
Oh, I'm sorry. Yeah, because we never changed that.
10, well, that works too. They're all interesting in their way.
Yeah, I'm sorry, but I did want that you still get the linear gradient.
The final configuration is independent of how you start.
Right. So the contour plots again are drawing lines over the areas of my simulation domain that have the same amount of chemical. And the gradient is decreasing linearly from 10 to 0, from 0 to 2, from 10 at 0 to 0 at 200. And since these iso contours are equally spaced, which what we should see when the system equilibrates is that the contours should be equally spaced between each other, because they basically divide the concentration in equal amounts of the field in equal amount of concentrations. Now we can also, let me see, in 10 minutes, yeah, we can definitely add a plot to measure, to visualize what the concentration looks like in the simulation domain. Let's do it in that same plot, in this same code. So what I need is first to add a window. That's, we'll use that, but not. We're gonna call it concentration versus position. X is gonna be my X position. And this is very similar to what we did with the pressure earlier. Let's call this concentration. And let's make it dots. And now I have created the window where I'm gonna plot my concentration as a function of my position. Now what I need to do is that I need every step, I need to iterate over the domain, and I need to measure at each location what the concentration is and then plot it. It's the same way we did with pressure earlier. So what I'm gonna do is first, because I know I'm gonna need it, I'm gonna add this erase plot. Then I'm gonna add the data points. But then what I'm gonna do now is I'm gonna iterate again over my x or my x plane. And then what I need to do is now I need to get what the concentration of my field is at the particular location. So I can go to CC3D Python, chemical field manipulation, get field value. Okay, but see how I have this variable here called field that refers to the name of the field that that I want to calculate. So I actually need that reference. So I come back to chemical field manipulation, and I need the reference to my field. And see, the way I reference my field is by using this property, and then notice that the field has to be in caps. So it's gonna be oxygen, right? So now this variable refers to my field, and now I want to calculate the value of my field at that specific location. So that location is gonna be X. It's gonna be at the middle in the y-axis. And now all I need to do is I need to add the data point to the plot. Again, I'm not plotting time, I'm plotting X. And now I need to plot the value of the field. Okay, let's just wait for people so we can play it. Just to wait for people to code. Is it clear what I'm doing here, right? I'm basically iterating over my X domain at half my simulation domain in the y-axis. I'm iterating over my X domain, getting the value of the field at each of my X locations, and then adding it to my plot. Okay, let's save and run. Okay, it might actually not be all caps. We'll have to figure that one out. Wait.
Try jogging the edge of your X term.
It's kind of like frozen.
Try joggling the edge of your X term down at the bottom. Make it redraw. Grab the corner, the bottom corner of the terminal. You have to do the bottom right corner. There. Pull on that. You may have to hover over it for a second to get it. Move it. All right.
Yeah, I don't know. Not necessarily sure where the CompuCell go.
Did it work for other people?
So someone hasn't finished, so I'm gonna show the code again. So just to make it clear, right? We are iterating over my X domain, over my X plane, and measuring what the concentration of the field is at that location and plotting it as a function. There you go. You can see it now. I don't know why is NanoHub doing this?
You have to move the bottom corner of your X term to get it to redraw. There. That's a bug in NanoHub's version of Xterm. There's nothing unfortunately, there's nothing we can do about it.
Right. So
It's
you
a
guys
bug
can
in
see now
Nano.
how the concentration is changing as a function of my location, right? And you also notice that it's always 0. It's always 10 at 0 and always 0 at 200. And then as the chemical diffuses in from one edge to the next one, it should the concentration increases, and then what we should see, as I was explaining, is that the gradient should be, the slope of this concentration should be linearly decreasing from one edge to the other one, which you would see either by an equal spacing of the ISO curves in this plot, or by a linear slope of the concentration as a function of the position. And I'm gonna stop there because it's 8, right? Do we go until 8? I might be confused because of the change in time zone.
No, you're done.
Okay, cool. Yep. So next time we'll quickly go over what happens to this when we add more terms into our diffusion equation. And we'll also quickly create membranes that that obstruct diffusion, and such that we can see what happens when chemicals try to diffuse through, for example, cell membranes that might be permeable or semi-permeable or impermeable, and how cells manage to take advantage of diffusion through membranes. Okay. Any questions before we part ways? Well, I appreciate everyone. I appreciate everyone's patience. I don't use NanoHub often for development, so if I run into a couple of problems there, I apologize. Promise that for next class I'll be struggling a little bit less with NanoHub. I'll have it more figured out. And I'll see everyone next week. Please do reach out if you need help with the homeworks or with your projects. We're more than willing to help you. Thank you.
Thank you.